I guess I want to echo before I begin Padma's sentiment that she shared with all of us about how I too have really enjoyed working with this group. It's been 10 years for many of you, and for the last 5 years for a bunch, and I'm going to miss it. It's been I've learned more in the last 10 years, I think, than for a long time. So anyway, it's been a pleasure to work with all of you. I am going to share my screen. And oops. How's that look? Is that the right?
Yes, that's good.
Yep. Okay.
Yep. Full screen.
So this is the final presentation for this project that we've been doing, and like everybody, and before I really dig into it, I wanted to give you a little bit of the biological motivation behind why we decided to do this. So Caenorhabditis elegans is one of the best studied animals in science. Had the first whole genome sequenced. There are tens of thousands of available mutants that you can just order from a stock center for the cost of shipping. And there are now collections of natural isolates that allow you to do precision medicine-like studies on C. elegans that are collected around the world. CRISPR is simple. You can grow them in controlled settings. It takes about 3 days to go from an egg to an adult. They eat bacteria. They don't hurt you in the lab. There are all sorts of useful things. And so the premise behind our entire project is that the wealth of genetic information that's been accumulated in C. elegans could really be useful in metabolite compound ID. And I should say there are a bunch of other model organisms that I think similar sorts of things could be said. We happen to love worms in particular. There are some analytical motivations that really drove the structure of our project. Probably the most important one is shown on the left. From Raphael Bruchweiler's lab in 2015, he and his team developed this thing that they called Summit. This is the simplest presentation of it. They've developed several different versions of this. But the idea is that you can take, if you can get a high resolution experimental mass spectrum and a good high resolution M/Z value, that you will be able to determine the molecular formula of a peak. And then you can find all of the structures that are consistent with that molecular formula through various computational methods, PubChem, other sources. And then you can go through and you can predict the NMR spectra from those predicted structures. So starting from the mass spec, you predict the NMR spectra, and then you record the NMR spectra from the same molecule, and you compare the matches, and the best one basically is a hit. So it's a great conceptual framework that we've really latched onto. There are all sorts of problems with it that I think Raphael knows well. We certainly know well by now, and you can imagine yourselves, but it's a really conceptually useful idea. The other motivation actually goes behind that, or over 10 years ago, I worked with Kenny Merge's group when we had isolated 3 diastereomers from a walking stick insect and collected NMR data. They were all produced in the same insect, and one insect in different ratios. And they have very similar chemical shifts. This is the overall structure of the compound, very different NMR chemical shifts. And we went to Kenny saying, if you guys are so good at calculating chemical shifts, can you tell us what the difference is between these diastereomers? And lo and behold, they could do it. We could actually predict which diastereomer was which just based on computation. And they also showed us why we didn't get the other possible form. So that was really remarkable. And just as a side note from the discussions that we've had yesterday, I just want to note that this actually the experimental data, I'm not showing the experiment, but you can look it up. It was just 1 microliter of spray from a single walking stick insect that gave us all of that information with a very high sensitivity NMR probe. So this is the team that we put together to do this Common Fund project. We have Facundo at Georgia Tech, Lauren at UF, Kenny, Eric Anderson, Franklin Leach, John Amster, Frank Schroeder, And we all brought different areas of expertise to this project. So it's been a real pleasure to work with everybody, all of the PIs on this. And this is something that many of you have seen several times. I could actually spend all half an hour talking just through this figure, so I'm not going to do that. But just to suffice it to say that we have had a lot of different samples that we have grown. We have had a lot of different experiments that we've conducted. So we've collected biosorting data. I'll show you an example of that in a minute. We've collected lots of forms of mass spec. We've done NMR, we've done LC, we've done different developmental analyses, a little bit of pathway mapping, and density functional theory calculations that I'll tell more about. And so lots of parts to this, but this has conceptually been a really useful framework for us to be thinking about. Something that I'm not going to be going into any detail, unfortunately, there just isn't time, is that we have had a huge amount of metadata that have been generated from this experiment. So we've grown bacteria, we've had a lot of different ways of growing bacteria, growing worms, harvesting worms, making different kinds of samples. And Abby has actually generated this thing that we call Google Sheets in our lab. That's just half of the platform, where she is able to record information in all of these different areas: biosource, the growth, treatment, preparation, data acquisition. And so this sheet makes it possible for experimentalists to add their data to as they're collecting it. And then Abby has actually made a back end that is incredibly useful and keeps it in a data structure where we can search. So we can actually say, for a given mass spectrum, what was the origin of the bacteria that led to that was used to feed the worms, for example. Over 15,000 entries we have right now in this really rich metadata set. So that's going to be for that. We're hoping to write that up in the next few months. So you heard Amanda talk yesterday in the lightning round about the study design. I'm going to kind of come back to that at the very end, but we did have to spend a lot of time developing how to make the samples in the first place. And so we have grown a lot of worms. Amanda has published this nice Jove paper to tell people how to do it. Very, very useful way of doing it. But we had to make some real decisions early on in the project. The biggest one was, what developmental stage do we focus on? We could have made the decision to synchronize worms and focus, for example, on the adult stage or the L4 stage. A lot of investigations do that. What we decided to do instead was to try to get a mixed population of worms on the plates and take what we get in all of them, so we have all of the stages. But that does complicate it. There was a lot of debate before we made that decision. On the next slide, I'm showing you. I've shown this slide before to at least the CIDC group about this. This is a time of flight of worms. So it's how long the worm takes to fly through the flow cell. And essentially, the longer it takes them to fly through, the longer the worms are. And so we can measure the length of the worms, and we can also record the extinction coefficient of the worms. So all of the points on this plot represents one plate that Amanda grew, and it shows us the developmental distribution for that plate. Amanda also has collected images of the worms in each plate for a select number of these things, to get an idea of the actual developmental stage, not just the size, but the stage. So that is a lot of data that we're still going through. So one question would be, does it really matter? Do we have to worry about developmental biology and all of this? And this is just an example of some data that we published a couple of years ago, right at the start of this project, where we took, this is just a different representation of the biosorting data that I'm showing on the left, but we separated this into 4 different worm size regions that we're calling it. And I'm just going to summarize a lot of data with this one figure on the bottom. We were looking at both glycomics and by LCMS and also NMR-derived metabolomics in this study. And what we found is that actually worm size, which is represented by the green hexagons in here, that they are very specific for not only the glycans that are expressed in the worms, but also metabolites, and that the metabolites and the glycans are very strongly associated with each other. So different worm sizes or worm developmental stages are really critical, we think, in this. So we have to keep track of all of this data. So one of the things that I'm most proud of, I think one of our best accomplishments in this whole project actually has been to come up with a way of making a reference sample. So actually looking back in our proposal, I proposed a rather naive idea. I knew that we needed to have a reference sample to go across the entire study. And I just said, well, we'll make about, a gazillion billion worms or something and just freeze them down and keep them forever. And Lauren McIntyre quickly corrected me and said, that's really a bad idea, Art. Not only is it going to be almost impossible to do that, but also what happens if your freezer breaks? What happens if it's a bad batch of worms that you make in a gigantic thing? So we spent a lot of time, and this is really the brainchild of Lauren and Gonzalo, pictured here, where essentially, and the significant part of this is that it's not just worms. You can really apply this to any kind of biological sample that you have. The idea is that you produce individual batches. So we grow up, like a plate of worms, and that they're represented by the letters going across. And then each batch, we make aliquots and freeze those aliquots, those individual aliquots, 1 through 6 in this case. And those are just frozen down. When we make our reference material, we will take a set of these batches together and a set of aliquots and mix them. So A through H will make one reference material. For example, the next time we get a reference material, it'll be B through I, and so on and so forth. So it's a rolling average. It's an iterative batch average that we have shown, Gonzalo showed with a lot of nice, careful measurements, that the variance of this material is significantly less than we would get if we just grew these separately. So we think that this is a good way to make a stable, long-term reference material for any kind of sample. Gonzalo and Bree, who I think are both on this call right now, we took this material to make our metabolite fraction library. And I'm going to take most of the rest of the time to tell you what we've been doing with this fraction library. This is essentially our version of trying to get Summit to work in our lab, the Brushweiler Summit. So the idea behind this is that we have samples that are grown on these big plates, and we extract the material from that we make IBAT material from them. We put them through a standard HPLC, either reverse phase or Hillick. I have to say that I'm always interested in what Charles is doing. We're not nearly as sophisticated in terms of chromatography as he was showing. I think at some point we've really got to get together and collaborate on some of these ideas, because those are good ones. But we are just doing basic preparative HPLC, collect fractions, we concentrate fractions, we do multiple injections so that we get a lot of material for NMR, and then we run these things. Well, we save the material, so we actually have material at the end of the day, and then we also run them on different kinds of mass spec. Carter mentioned getting ion mobility data. In Facundo's lab, we collect NMR, and we have these different data matrices. And then in a study like the ones that Amanda told you about yesterday, we have a whole study here where it's more of a traditional profiling kind of case. We collect the same kind of data, but now we want to try to bridge the experimental study with the fraction library. So this is a little bit more detail about what we actually do. We start with this reference material. We make 2 different extractions, polar and non-polar material for all of our analytical platforms. We fractionate. Typically, we collect about 100 non-targeted fractions. We just time them. And then on each fraction, we do MS2, MS1, 1D NMR, and then right here it says we targeted some fractions for 2D NMR. We've now completed the 2D NMR data collection for database annotation, the basic 2D NMR. We can do a lot more than this afterwards. And then we put these things through various database matching filters and see what we get. So we're using Sirius, GMPS, and then we also are using the Buschweiler-Colmar suite of matches, and we try to see how they correspond with each other. So this, what I just said is shown here again pictorially, where we have fractions and time. Again, we monitor the data with UV. We don't have a mass spectrometer on the HPLC. We would love to get that, actually. That's one thing that would help us a lot. But we're collecting individual fractions. It's actually a big advantage if these are not pure fractions in us. And so having a fraction run across different tubes is quite good, or a peak going across different tubes really helps us. And so we can also do more orthogonality, as I indicated, but it is key that it's untargeted in this case. And then essentially now we have a new dimension, fractionation time or fraction number, that we can put into our data analysis. So our data are shown here, and on the right are 1D NMR spectra of the fraction library, and you don't see nearly the amount of detail that's actually in here, but so these are 1D NMR spectra separated by fraction, and then over here is the LCMS separated by fraction. And in this case, we're using the same chromatography. You can see by a diagonal sort of structure there. We try to get the chromatography fairly matched. And so now I'm going to show you where we have gone trying to match everything together. And this part, we're still working on it. We think that we have the annotation part solved, but there are a lot of subtleties that we didn't fully think out. And I'm more excited than anything about this technique, but there's a lot that we still have to figure out. So I mentioned in our group discussion yesterday, at the start of the day, that one of the problems, in my opinion, in NMR has been the fact that we are adamant about the fact that we don't have to do a lot of sample preparation, that we can just put a sample in and we can collect data. And on the left, on A, this is an unfractionated sample from our study that Amanda told you about, one of our just experimental samples. And you can see there are a lot of peaks, but there's a lot of overlap. And so this is what normal NMR metabolomics is oftentimes working with, if it's just 1D. And so when we put it through the HPLC, in this case, I'm expanding what's in this box. And you can see this is just the sugar region. And you can see clearly there's trehalose that comes out at fraction 47. We have glucose that comes out at fraction 41. Those are really heavily overlapped. I mean, they're obviously both very similar sugars. And you can see more of just the region of the different traces across these fractions from 46 to 59 in here, and you can see the degree of overlap. I mean, this region is pretty crowded anyway, but it's pretty remarkable actually to see like going from 46 to 47. You can see little isomeric shifts. There's something going on there with unique compounds that we would not have seen before. So that's one thing, is that we're getting a lot of resolution of overlap. The other thing is that we're actually concentrating the sample. So we're doing multiple injections, and we can do as much as it's auto inject HPLC. And so we can concentrate our sample so we can make up some of the loss of, or some of the lower sensitivity that NMR has, and get more material in there. So then what we can do is essentially do like extract out get the NMR peaks or the mass spec M over Z values over fraction numbers. So this is an example up here of the chemical shift at 0.9529 ppm extracted over all of the fractions, and it has this elution profile, if you will, for that one NMR chemical shift right here. And then we can look in the mass spec again, extracting over the HPLC fractions, and we can find similar profiles. And we can say, those are probably they probably belong to the same compound. In this case, just leucine as a simple example. We can then go to that fraction, and so, well, one of the issues is that as long as we get rid of, do a good job getting rid of all of the isotopes and the fragments and the thises and the that's and the mass spec, then we're going to have one molecule for each m/z value and retention time value. And but in NMR, we're going to have multiple chemical shifts. So we then can we can look at the experimental sample and say, where are those chemical shifts? So up here are all of the PD1074s, the anchor. If you remember from Amanda, she talked about the anchor strain. So we have about 40 different spectra collected essentially under identical conditions here. And we can ask the question, what is statistically correlated to this peak, 0.9529? And this is Stocksy that Nicholson's lab has developed, and this is what we get. Useful, but maybe not so informative here. We know what this one is. It's not that hard. But then what Gonzalo did is he took these dark red peaks that have a high correlation value, and he looked at the covariance of those peaks across all of the fractions. And so this is where those peaks localize. Again, fraction 41, 42, we can take traces out and we can find different peaks across those fractions and see what we're getting in there. Then we can do 2D NMR on these fractions. This is an example of a toxi and an HSQC. We can get matches. In this case, again, it's pretty simple. It's leucine, but we can get the database match to these things. It's quite accurate, quite reliable. And then we can look in those, we can look for those fractions. We put the fractions through GMPS, through the LCM SMS, and all of the fractions we can put into GMPS, and then we can color code the nodes by the fraction number. So in this case, it's 40 and 41, and we can actually go down and find what GMPS is telling us. And we can start to relate the mass spec with the NMR. So there's a lot of bridging that we can do with this. We've taken, this has to be updated now. We have a lot more data, but it still is in the analysis stage. But we've looked at 5 fractions with 2D NMR. Now we have all of them done. We just have to do the analysis. We've matched them to Colmar. We've taken the positive mode data from Helic. We now have the negative that we're adding to this before we publish it. But we're looking for a unique hits in each of these. And surprisingly, that there are very few compounds, only 3 or 4 that are actually present in all of these different database searches. There are a lot of single ones that we get, and so we can start to figure out. We can use this, we think, to help us come up with a different annotation and matching scheme as well, like we talked about with Tom. Now, in the last couple of minutes, I just want to tell you about what we are doing with Kenny's group to improve the NMR chemical shift. Essentially, Susanta and Kenny came up with a really slick way of quickly, or more quickly than we could do before, calculating NMR chemical shifts for both proton and C13. They could get about 0.1 to 0.2 ppm error in proton and around 3 ppm error for C13. So this is a fairly fast workflow, and we can start to think about merging the bridge that Charles alluded to, merging these two sides through these computational means. Now, you heard from Carter and from several people about CCS calculations and the use of ion mobility. The problem that we still have is that there are a lot of different combinations, and it's really tough. There's still hundreds of compounds that we could actually go through and do searches. And so what we're now doing is we're using CCS as a filtering step on top of this. And Susanta and Kenny's group have also made a CCS prediction pipeline. This is featured in this POMIX web portal that I highlighted in the quantum mechanics working group that Keo and Susanta have put together. Essentially, you put in a SMILE string, you get out a CCS value. And so we're hoping that this can be used as a filter to improve our ability to match everything together between mass spec and NMR. And then finally, this is almost the end, I promise. You heard about this study from Amanda. I'm not going to go into it, but this was really the warm up. And this is, and it's both, it's harder to do than we thought it would be, but also COVID came and it really slowed up a lot of the steps in this. But we're nearing completion of this study. You heard about the anchor study. I'm not going to tell you more about that, but we're applying exactly the same pipeline now for the next year during this no-cost extension, year 5, to the TOR pathway. So this is where we're really going to be able to investigate the genetic aspects to what I was talking about at the start. So each of these genes, we have knockout strains. We have them in our freezer. We've grown the samples in the same anchor design that we used before. We have the same IBAT reference material. Data collection is underway right now. All of the analysis pipelines are built, and we're also going to be adding RNA-seq to these TOR mutants to try to really put it all together into one coherent package. That was actually thanks to Dean Jones in one of the early face-to-face meetings for that idea. And so with that, I'd like to thank NIH, all of you who have been so helpful and supportive over the years. I want to thank my team. This is one of our first face-to-faces since COVID. These are the folks that have done all of the work, Franklin and John Amster at UGA, all of the consortium folks, and then all the collaborators have been just amazing on this project. So with that, I thank all of you as well.
Great. Are there any questions?
Yep, I have a question if okay.
Okay, great.
Great presentation, Art. I like the worm study. I like that as a reference material, and I like the whole approach. Plus I always like to say Art's got worms, so that's always fun. Is it practical for NMR spectra to try and do like a spectral similarity score against a pure reference spectra? I know interpretation of NMR spectra is complicated because it's got much more information through bonding and so forth contained. But what are your thoughts in terms of like full automation of just simple, like say cosine similarity matching?
That's a good question, and I'm not sure that I know the answer. I think that Gonzalo mentioned in question answer yesterday this smart NMR thing that Dorenstein's group have created, which essentially takes HSQC data, which is routine for us, and we always collect that, and then finds it's an automated way to sort of look at your sample. I don't know how well that's actually going to work, but yeah, I'm not sure. It's an interesting idea. We do have pretty good luck matching databases if we have the spectra in the database to match. But the NMR databases are not as complete as mass spec databases for sure, but they're pretty stable and it's easy to match once we have the compound.
Okay, thank you.
Okay, other questions or?
Or real quick, thanks for a great talk. I was just curious, that first slide you showed where you were talking about going predicted NMR spectra to matching up, and you alluded to problems, and then you told us some other interesting ways that you're lining things up. I'm just curious, when you get to that approach, how often are you successful at finding, for not knowing that much about NMR, but unique NMR peaks that can definitively link, a predicted structure to something that you can see in your NMR that you're have you tried that out with the data that you've been collecting, and how does it look?
Yeah, so we actually spent months trying to do exactly that, trying to take unique peaks and get them connected to what we think is the right M over Z value or MSMS value. One of the problems is that a tool that we use quite often in our lab, I mean, I mentioned STOXY, which is just statistical correlation spectroscopy. That's That's Nicholson's term for within an NMR spectrum, you get correlations. You can do that outside of an NMR spectrum. They call this SHI, statistical heterospectroscopy. We use SHI a lot. We love that. And we tried to do it with this fraction library, but the problem is that you don't have, you either sort of have it or you don't. You don't have that many data points. And so it's really hard to get to believe any correlations that you get. We just don't have the kind of data structure that make that possible. And then to combine it, we have positive mode, negative mode, mass spec data. It's hard to bring all of that together and to try to find good ways of matching. And then we probably have, I don't know, 50 or 100 compounds in each fraction. So it's been a little more complicated than we had originally anticipated, even though it's a lot simpler than what Raphael initially put forth in his summit paper. I still think that we can find what we think that this database matching and increasing the confidence of annotation is it has a lot of value to it, but I think that we can also, with more computational chemistry, really bridge that divide.
Thanks.
Well, great. This great presentation. This has been a great meeting this last couple of days.